Right, good evening. Good evening. Hello. How are you? Am I audible? Can you hear me? My students, how are you? Komanda, how is the evening? Is it raining here? People, there is a light shower here. How about your area? It was thundering and shower now. It has stopped. How about in your area, people? Is it raining there? Huh? Is it raining? A bit, ne? Santosh say a bit, and Zeenam says, no, sir, not so good, sir. Hari, good evening. Hello, Sama. How are you? How are you, people? How are you? Great evening. Yeah, fine. Hope you are fine too. Yes, straight from Kurunagala to online platform, right? So, we have to discuss paper number 6 today. Paper number 6. Paper number 6. Find that paper 6, people. I have shared it to the online students. It's there in the website also. Did you answer paper number 6? Paper number 6. Right. Normally, people, now we go on a reverse way, the reverse way, so that we start with the essay questions and then we go backward. Right. That is, I think, a better way of discussing this paper. Right. Better way of discussing the paper. Reverse discussion. Right. Otherwise, we always miss people the more important essay part. Ne? So it's better to start with the essay section. Essay part. Right, people, essay questions. I'll give you this marking scheme on background. Marking scheme on background. People later. On Saturday, I'll give it to you. Yes. Right. Question is, write an essay on the mechanism found. Write an essay on the mechanism. Write an essay on mechanism found in C4 plants for fixation of carbon dioxide molecule and synthesis of organic food. Right. This is regarding the C4 mechanism. You learn C4 mechanism, neither. You know C4 mechanism. So where carbon dioxide is initially fixed by people PEP, and PEP carboxylase is the enzyme. This enzyme is special because there is no oxygenation reaction in it. And then carbon dioxide is going to be fixed and then form people 4-carbon compound. What do you call that? So can you send the paper to the chat box? It's given. Shall I send it once again? It's there. Right. So you have to say that oxaloacetate is produced, oxaloacetate is converted to malate, malate, aspartate, and then people, yes, and then this malate is going to move into the bundle sheath cell from the mesophyll cell, goes into the bundle sheath cells, and inside the bundle sheath cells, malate is converted to pyruvate by releasing carbon dioxide. So decarboxylation occur there. Decarboxylation occur, and this decarboxylation carbon dioxide is going to participate in the normal Calvin cycle which happen inside the bundle sheath cell. So you have to like little bit describe this Calvin cycle which occur there too. Calvin cycle occur, this carbon dioxide absorbed, and then this is going to transform into G3P, the final product. And then the pyruvate move back into the mesophyll cell and then converts into phosphoenol pyruvate with expenditure of what we said ATP. With expenditure of ATP, this conversion is going to happen. Let's check the marking scheme for these people. Let's check the marking scheme for that. These are the points that we consider. So if you have already answered the paper, please upload it to the website, right? Upload it to the website. We'll mark it and give you the answers. In the mesophyll cells of C4 plants, carbon dioxide is initially fixed by phosphoenol pyruvate using PEP as carbon dioxide receptor. The resulting oxaloacetate is composed of 4 carbon atom. Oxaloacetate is rapidly converted to more stable C4 compound, malate, or as you can say aspartate, malate or aspartate, that diffuse into the bundle sheath cells. Carbon dioxide is released by decarboxylation and release carbon dioxide is refixed by Rubisco, which is exclusively operate in the bundle sheath cells of the C4 plants. Rubisco present. Chloroplasts found in mesophyll cells are different from anatomy compared to the chloroplasts in the bundle sheath cell. You learn, people, chloroplasts in the bundle sheath cells. Chloroplasts in the bundle sheath cells are less differentiated for the light reaction, more differentiated for the Calvin cycle, right? And it contain less thylakoids. And you know, people, some specialty is available here in the thylakoid. What sort of difference present? Can someone tell me the difference? About the PS1 and PS2 availability, we see some difference. What is that? The PS1, only PS1 is available. Only PS1 is there. So the grana of the mesophyll cell chloroplast are large and highly differentiated into light reaction. Bundle sheath cells possess very few less differentiated grana or grana absent. Grana absent. They contain only PS1 and not PS2. ATP generated only through the cyclic electron flow. This PEP carboxylase enzyme is much more efficient than the RUBP carboxylase enzyme. Calvin cycle occurs in the bundle sheath cells. Calvin cycle of photosynthesis can be described in 3 steps: carboxylation, reduction, regeneration occur there. The carbon dioxide receptor is 5-carbon sugar, ribulose bisphosphate. Addition of carbon dioxide into REBP is called as carboxylation. The enzyme involved in this reaction is REBP carboxylase. So this is a description regarding the cycle, Calvin cycle, which occur inside the bundle sheath cells. I think this sort of a question, people, you better describe very briefly about the Calvin cycle too. The first product of REBP carboxylation is 6-carbon molecule, 3-PGA, unstable compound, and break down into 3-PGA. This is the first stable product. The enzyme Rubisco REBP carboxylase oxygenase is present in large amount. 1,3-bisphosphoglycerate will reduce to glyceraldehyde 3-phosphate. Enzyme catalyzed reaction utilizes in ADP and ATP. REBP is regenerated. This process uses energy from ATP generating the light reaction. Glucose is later synthesized. That part, I think I have mentioned that. The complete C4 pathway you have to mention here. C4 pathway completely you have to mention. That is, the pyruvate is converted back into phosphoenol pyruvate with expenditure of ATP. I think that point is missing in the market scheme. I'll include that to the final market scheme. Okay. It says carbon dioxide is prefixed. Yeah, and then the rest of the part is like description on the C3 pathway. But anyway, the last part, I think I need to add one more point for that. About 2 points I have to add further. Right, other information are there. Then people, we go for the second question. Second question, briefly explain the significant features of seedless vascular plants. Significant features of seedless vascular plants. Seedless vascular plants, what are the phyla comes under that, people? Seedless vascular plants, when I say, what are the phyla comes under that? Come on, tell me, what are the phyla under that? What phyla comes under that? Come on. Come on. Hello? What phyla? Lycophyta. And, oh, Bryophyta. Oh my God. Seedless vascular plants. Seedless vascular plants. Yeah, Pterophyta and Lycophyta only. Ne? Pterophyta and Lycophyta only. So that you have to mention here. And then when we talk about significant features, what significant features we discuss here, people? Significant features. We divided these significant features under three main points. We have discussed these three main significant features. What are those three? What are those three main significant features, people? Transportation through xylem and the phloem. That is correct. The first one. What is the second one? Evolution of roots. That's very good. And what is the third one, people? Evolution of leaves, right? So the information relate with those three. You have to describe. When we say transport through vascular tissues, you have to say about the xylem arrangement and the phloem arrangement little bit. So that's the marking scheme. That's the marking scheme, people, right? So look at this. Vascular plants have two types of vascular tissues, xylem and the phloem. Xylem consists of tracheids, fibers, and parenchyma cells. They conduct water and minerals. Cell walls or tracheids and fibers are strengthened by lignin. This may facilitate them to obtain high amount of light for photosynthesis, because this helps the plant to grow taller. Right? Having vascular tissues helps the plant to grow taller. Right? These permit the plant to grow tall. So it helps in the spore dispersal and effective photosynthesis. Phloem is a tissue with cells arranged in tubes. It distributes sugars, amino acids, and other organic products among different parts of the plant. Evolution of roots are organs that absorb water and nutrients from the soil. They help to anchor the plant and allow the shoot system to grow taller. They also replace in the rhizoids seen in Bryophyta. Root tissues of living plants resembles the stem tissues. And that's like a special point. So this one, right? That's like a special evolution detail. Evolution of leaves. Evolution of leaves. Evolution of leaves. There are two types of leaves, microphyls and megaphylls. Right? The lycophytes are having microphyls, and then pterophytes are having megaphylls, right? So microphylls are single veined and smaller in size. Microphylls small, and then you learn, people, vascular tissues are unbranched too, in microphylls. Megaphylls are large and flattened with branch veins. Leaves with branch vascular tissues increase the surface area for efficient photosynthesis. Sporophylls and spore variation present. Modified leaves that bear sporangia are known as sporophylls. Most seedless vascular plant species produce one type of sporangia and one type of spores. Therefore, they are known as homosporous. Homosporous. But Pterophyta is homosporous. Pterophytes are homosporous, right, people? And then you learn, people, among the lycophytes, one is homosporous, the other one is heterosporous. Which one is homosporous? Which one is homosporous? I'm asking from you. Which one is homosporous? Club moss. Thabana Jaiprakash says club moss. What is club moss? What is club moss? What is the genus name of club moss? Who's going to tell me? Right. There are a lot of people to say Lycopodium. Yeah, that's correct, right? Lycopodium, homosporous. And then heterosporous, the spike moss, or what do you call the Selaginella is heterosporous, right? Good, neither. And then people, most seedless vascular plants, some plant species produce two types of sporangia, produce two types of spores. They are called as heterosporous and called as megaspores and microspores. Megaspores develop into female gametophyte and microspores develop into male gametophyte. Right, people? So that's the part A. Part B, explain how lycophytes are different from pterophyta. Explain how lycophytes are different from pterophyta. Lycophytes are different from pterophyta. right? What do you say, people? Pterophytes, ferns need like Neprolepis. And then you learn people Neprolepis is both are seedler vesicle plants, right? You can see that the Neprolepis, um, the underground stem is there, but in Lycophyta we can see that the ground hugging stem is present. Lycophyta, we said people strobilus present, homosporous or heterosporous condition is there. Neprolepis, we said always homosporous condition. Sorry, pterophytes are having homosporous condition, right? And then you learn people the gametophyte. Gametophyte, we said that can be monoecious or dioecious when come to Lycophyta. But pterophyta, we said that the gametophyte is monoecious, right? Heart-shaped gametophyte present in Neprolepis, right? So that makes a difference. And so people, these are the main points I have taken. Lycophytes produce upright stem and ground-hugging stem in Pterophyta. The rhizomes, they produce leaves called fronds. Lycophytes have strobili, homosporous or heterosporous in Pterophyta. No strobili, no strobili. They are homosporous. In some Lycophyte species, tiny gametophytes live above the ground and they are photosynthetic. Others live underground and they nourish by symbiotic fungi, but pterophytes have bisexual gametophytes. And then always people remember, I have not followed that neither. When you write the scientific names and you got to underline that scientific name. Right, well, coming to the third part, that's about writing short notes. Write short notes about galls in plants. Plant galls, we said people, these are the tumors that grow in plants. And you learn people, gall is a clump of cells. And these galls are actually produced due to the loss of the balance of the cell division. Some hormones like auxin and cytokinin promotes this cell division process. And especially people, when the plant tissues are under certain attack by the gall causes such as virus, what else? Virus, fungi, insects, mites and all. When they damage, the cells lose their normal control and they go on forming a cell clump that we call as a gall. And yeah, so galls occur under uncontrolled meiotic division. The plant division controlled by maintaining proper balance between the plant growth regulators such as auxins and cytokinins. When this balance is lost, plant causes undifferentiated mass of cells. Galls are bumps and growths that develop on different parts of the plant after being invaded by some very unique organisms. We call gall causes virus, fungi, bacteria, insects, and mites. Bacteria also there. People in root nodules are also galls, right? Present in leguminosae plants, you learn it. The gall causes some way attack and penetrate the plant growing tissues and cause host to reorganize the cells and then develop into abnormal cell mass. Then people, the second one, mechanism of enzyme action. Mechanism of enzyme action, we learn people, enzymes are having a particular site to which the substrate go and bind. The enzyme reactants are the substrate. So when the enzyme and the substrate bind, they form the enzyme substrate complex. And then this is going to transform into products. Right? And then you learn people, enzymes are not always complementary to the substrate. As the substrate come and interact, there is a induction of the enzyme active site, making it more complementary to the substrate than helping in formation of the enzyme substrate complex, right? And then form the products, right? So this is allowing very close binding between the enzyme and the substrate, helping for the product formation. And so this allows the product formation. Read that part, people. The reactants on which the enzyme acts is referred to as the substrate. The enzyme bind to its substrate, forming enzyme substrate complex. Catalytic action of the enzyme converts the substrate to products. The specificity of enzyme result from its shape. The substrate bind to specific region of the enzyme called the active site. Active site is formed by only a few amino acids. That's an important point. Yeah? Active site is formed by few amino acids. Right? Other amino acids are maintaining the shape of the enzyme molecule. The shape of the active site of the enzyme is complementary to the shape of its specific substrate. I think I had to go to other page. Here. Right. Then shape of the active site of the enzyme is not always fully complementary to the substrate. The interaction between the substrate and the active site slightly changes the shape of the active site so that the substrate and the active site become complementary to each other. This is called induced fit. I forgot to mention that, induced fit. The tight fit brings the substrate molecule and the active site close together, also ensures the correct orientation and catalyze the conversion of substrate to product. Thereafter, the product depart, and then the enzyme is now free to take another one. Yeah. Right, people. Then regarding polysaccharides, part C, they are macromolecules. They are biopolymers. Polysaccharides are made up of few hundred to few thousand monosaccharide subunits. They are non-crystalline, water insoluble, and not considered as sugars. Some polysaccharides are storage compounds, where others contribute to the structure of the living organism. Based on the function, they are categorized as storage and structural, right? And then example, storage, starch and glycogen. Example for structural, cellulose, hemicellulose. And then structural, we can take cellulose, hemicellulose, pectin. Based on their architecture, categorized as a linear, cellulose and amylose, some are branched. So altogether, people, 40 points. Altogether, 40 points for part C. So we can divide marks accordingly. So you submit your paper, we mark it, and then hand over back it to you, right? So we'll do that, right? So you know how to create the folder and upload it to the folder. You can hand over it on Saturday also, but then people, if you can upload it, it's better, right? We promote uploading, right? For the 2027 batch, we promote the submitting in the physical class for the 2026 batch, right? So we can collect it and then correct it and then discuss their weaknesses, right, in the physical class, right? So whatever, one of the option you can use. I think people uploading is better for the 27 batch, right? I'll promote more that. And then people, the structured essay paper. Structured essay paper will start with second question. Structured essay paper will start with the second one. Write generic names of seedless vascular plants. Second question. Write generic names of seedless vascular plants. What are the generic names of seedless vascular plants? Generic name of seedless vascular plants that share more recent common ancestors with the, write the generic name of a seedless. I think this is wrong. Write the generic name of the seedless vascular plant that share more recent common ancestor with the plant with seed plants. This is not Lycopodium. It has to be Neplolepis. Neplolepis. That's Lycopodium and Selenula is wrong. Neplolepis, right? I'll correct it. Okay. And then second one, seed plants can be divided into two groups based on the special features that feature. Seedless plants can be divided into two groups based on the special feature. Seed plants can be divided. What do you call people? Open seed or naked seed plants and the angiosperms. This is correct. Naked seed plants and angiosperms. Place a tick on the appropriate column. Let's find that. Tick on the appropriate column. Tick on the appropriate column. Tick on the appropriate column. Seed without fruit. Seed without fruit. We'll take cecus. Seed enclosed within chamber. Seed enclosed within chamber or rhizae. Then heterosporous. Heterosporous, you can take people Selaginella, cecus, and Oryzae, all three. Ne? What is Oryzae? What is Oryza? Oryza is rice. Ne? Oryza, all three. Ne? And then flagellated sperms. Flagellated sperms are present in Selaginella, and you can take for cecus. When come to the other seed plants, like in Pinus and in Oryzae, people flagellated conditions are not present. And then people, presence of endosperm. Endosperm is present in cecus and Oryza. So we'll take it like that. Okay. Give special features found in their success on terrestrial life. Give two special features found in the seeds for their success on terrestrial life. What are the special features present on seeds? Seeds for success on terrestrial life, people, they have an endosperm. Ne? Seed coat is there, right? Protective coat present and endosperm present. Those two features you have right there. Indicate two triads that can be shared by sporophytes of the bryophyta and vascular plants. Indicate two triads that can be shared by the sporophytes of bryophyta. Sporophyte of bryophyta and that of the vascular plants. You learn, people, sporophyte of bryophyta has stomata. They are tiny pores present on the capsule, and that can be seen present among the Pterophyta, tune, and other vascular plants. So that we will take presence of stomata as a point. Part B1, it is believed that the mechanism of kingdom, it is believed that the members of the kingdom plant were evolved from group of chlorophytes. Give three characteristic features which are present in terrestrial plants, not in chlorophytes. You learn, people, the chlorophytes are green algae, and then the promotion into plants, having whorled spores produced in sporangia, having apical meristems, having what else, people? Having multicellular, the sex organs, having multicellular sex organs, and having a dependent embryo. Those four features we can take as special features that had evolved, right? Read the question once again, people. It is believed that the members of kingdom plant were evolved from group of chlorophytes, the green algae, and then give three characters which are present in terrestrial plants. Okay, yeah. Algae, people, they don't have whorled spores produced in sporangia, no apical meristem, no multicellular sex organs, and then people, no dependent embryo. They don't have even an embryo stage. They don't have an embryo stage. So these are the special features that we can take. Right, those are the special features that we can take here. Right, people. Then moving to next question, part B, second question, state the phylum of seedless vascular plants that has closer relationship to bryophyta and name two genera. State a phylum of seedless vascular plants that has closer relationship to bryophyta. We had a similar question before, State a phylum of seedless vascular plants that has closer relationship to bryophyta and name two genera, that is Lycopodium and Selaginella. Lycopodium and Selaginella. Right. Next one. Complete the following dichotomous key to distinguish plant species given below. I think most of the students, they have skipped this question. I checked the last time, Saturday. Neplephis, Pogonatum, Marcantia, Hibiscus, Pinus, Paddy. There is a small weakness here. Neplephis, I had underlined them. Pogonatum, Marcantia, Hibiscus, Pinus. Right, all scientific names must be underlined. And xylem present. If you take people, xylem present. How to take? If you draw uh, a plan for this dichotomous key, xylem present. So these plants, Neplephis, Pogonatum, Marcantia, Hibiscus, Pinus, and Paddy. If you take these plants, xylem present. This is our plan, neither. Xylem present. Xylem present, you can take Neplephis, you can take Hibiscus, Pinus, Paddy. Neplephis, Hibiscus, Pinus, and Paddy. Vascular tissues present. Then xylem absent, Pogonatum and Marcantia. Pogonatum and Marcantia. So we take this as number one step. This is A and this is B. Then xylem absent, xylem present. This goes to number 3 step. Xylem present. Xylem absent goes to number 2 step. Now, thalloid body is available, people. From these two, Marcantia is a thallus body. Thalloid body. What is that? Marcantia. Now, you can't take, for these plants, you can't take a thallus because they have vascular plants, stem, root, leaf, differentiation present. Right? So, this is A, 2A, this is 2B. 2B, stem present. Stem present, that is Pogonatum. Pogonatum has a stem. So, this is step number 2. Then this one is step number 3. Step number 3, 3A. What is 3A? Homosporous. Which one is homosporous? From Neplephis, Hibiscus, Pinus, Paddy, which one is homosporous? Only Neplephis, This is then homosporous. Neplephis. So this is now gone, Neplephis. So then people, we take heterosporous. Heterosporous, then you can take Hibiscus, Pinus and Paddy. And then people, that goes for step number 4, Step number 4A and 4B. What is 4A? Cone present. Which one is having a cone? Pinus has a cone. So cone present, you have Pinus here. Cone absent, then you have 2. Hibiscus and Paddy. Cone absent. Then the last step. Pollen grain contain 1 pore. Which one is having 1 pore? Paddy. Why? Monocot. 1 pore. Pollen grain contain 3 pores, that is Hibiscus. Right? So you can do that easily. Right? So this is the answer here. This is the answer. How many of you have answered this correct, people? Please raise your hand up. How many of you got this correct? Raise your hand up. How many of you got this correct? Come on. How many of you got this correct? Nobody? 4 people got it correct. 5. 5 people. 5 people got that correct. So that's good, ne? Right? Those people are like, right, the ones having more potential for the medical faculty, ne? Right? Other people are going for the other faculties, veterinary, the agriculture, right? And then people are science faculty, ne? Huh? So that's good, huh? So if you don't like, I told you, ne? If you don't like the science faculty and the other faculties, say the agriculture and all, so you better work on this part a little harder, right? And avoid this danger of letting you go to a faculty where you have to learn all these things repeatedly again throughout your life. Haridha, I have warned you enough, huh? So I have warned you enough. Right, this is it. Part C. Part C. Right, part C, what are special features of flowering plants? What are the special features of angiosperms? Presence of flower, production of the fruit. State function of fruits in angiosperms. Fruit protect the seed and helping in the dispersal of the seed. State 2 classes of angiosperms, monocotyledonous and dicotyledonous. State 7 differences between the above mentioned classes. People, this is one of the exam question also. Recently they have asked to differentiate between the above mentioned classes, right? Monocot and the dicot, differences you have to write there, right? Monocot and dicot differences you learn, neither. Monocot is having one cotyledon in the embryo, 2 cotyledon dicots. Monocots are having the stem anatomy differences. The vascular bundles are scattered and no cambia, and vascular bundles are arranged in a ring and cambium present when come to a dicot. And then people, you learn the leaf arrangement, some differences. Monocot leaves are having parallel venation, dicot leaves are having reticulate venation. Roots differences are there, adventitious roots are there, fibrous roots, ne? And then in the monocot plant, dicot has taproot system. And then the flower, some difference are there, trimerous flowers, tetramerous or pentamerous flowers in dicots. And then the pollen, and then the differentiation of the calyx and the corolla, some difference are there. Perianth is present in monocots. Perianth not differentiated into calyx and corolla in dicots. And then you can see pollen grain with one opening in monocot, 3 openings in dicot. So many differences are there. So many differences are there. So expecting that as the answer for that question, expecting that as the answer for that question. And then people moving to first structured question, moving to first structured question, right? Structured essay answers. What is a photosystem? Right? Question, what is a photosystem? You learn, people, this is a special complex where the organic molecules and the other pigments are packed neither. These are the chlorophyll molecules and other organic molecules, and proteins are organized into complexes in that the thylakoid membrane of the chloroplast. 2 marks given for that. What is the structural arrangement of the reaction center, not the center, center, reaction center of the photosystem? What is the structural arrangement of the reaction center? Pair of chlorophyll A molecule and primary electron acceptor present. So I think pair of chlorophyll A molecules, the what do you call the key pigments, either P680 or P700, 2 pigment molecules are arranged in the center, plus primary electron acceptor is available in the center complex, right? What is the wavelength? What is the wavelength of light absorbed by photosynthetic pigments? What wavelength of light is absorbed by photosynthetic pigments? Violet, blue, and red light. Now, if they specifically ask for photosystem, the center complex pigments, P680, P700, you have to like specially mention, but generally photosystems, no, right? Don't say particular wavelength. Now that particular wavelength comes to the center complex pigment. Otherwise, the photosystem absorbs in a whole range. Both photosystem 1 and photosystem 2 absorbs in a range. Center complex, all the pigments are now by fluorescence, they send a particular wavelength, 680 in the PS2, 700 nanometers in PS1, right? So don't write that as the answer for this one. Okay, so we write people here violet, blue, and red. And then people complete the following table regarding the type of the electron flow in light reaction, prime electron donor, final electron acceptor, and products. Linear. Complete the following table regarding the type of electron flow in light reaction. How the question given to you? Let me check that, people. Right, you have to write. Complete the following table regarding the type of electron flow. There are two electron flows, neither. What are those two electron flows? One is called as a linear electron flow. The other one is a circular, neither. Cyclic or circular, what we said. Cyclic electron flow, neither. Cyclic electron flow. And then primary electron acceptors of PS2. And then the circular electron flow, primary electron acceptor of PS1. PS1. So there is a small typing mistake. And then people, final electron acceptors, NADP plus reductase enzyme is a final electron acceptor. Am I wrong here? Final electron acceptor is NADP plus, not reductase. NADP plus. And products are ATP, NADP, H, and oxygen. So isn't it NADP plus for the final electron? So yeah, that's my mistake. And then final electron acceptor here is PS1. And then the products are going to be ATP. Right? Products are going to be ATP. That's good, right? So small errors are there. So we'll fix them, no? Cyclic electron flow. At PS1. Right. Next one is, what are the global importance of photosynthesis that you can write? All life depends on directly or indirectly on photosynthesis. Fulfill both carbon and energy requirement, provide oxygen for respiration of aerobic organism, maintain oxygen carbon dioxide level, production of fossil fuel. These things you can write. No problem with that. Go to part B. Where does Calvin cycle take place in the chloroplast, people? Where? Stroma of the chloroplast. Happens in the chloroplast stroma. And then what are the three main steps of Calvin cycle? You know what to write. Carboxylation, reduction, and regeneration. How does increase of the oxygen concentration in mesophyll cells affect? How does increase in oxygen concentration? We learn, people, in C3 plants, when oxygen concentration increases, the oxygenation reaction increases, reducing the photosynthetic efficiency by 50%. What do you call this process? Photorespiration. Photorespiration. So that is what you have right here. Right? REBP bind with oxygen and the oxygenation reaction occur, reducing photosynthetic efficiency. 3 points are expected here. Name 2 nucleotides that are used in Calvin cycle. One is NADPH, you can take. The other one is ATP. But you know NADPH is a dinucleotide. This is dinucleotide, neither. Dinucleotide. But you can take it as a nucleotide. And then ATP is also a nucleotide. Then what is known as action spectrum? It is a graph showing effectiveness of different wavelengths of light stimulating photosynthesis. Name the type of photosynthetic pigments that prevents the formation of reactive oxidative molecules, carotenoids. That is carotenoid. Name two types of cells in which carbon dioxide fixation occurs in C4 plants. One is mesophyll cell, the other one is bundle sheath cells. And state the carbon dioxide receptor mesophyll cells, phosphoenol pyruvate, bundle sheath cells, REBP, and carbon dioxide fixing enzyme PEP carboxylase and rubulose bisphosphate carboxylase oxygenase. How do two types of cells stated in C above reconnect tightly with one another? They tightly reconnect with one another by people having high number of plasmodesmata. This is important. Mention high number of plasmodesmata. Right? High number of plasmodesmata are there. So those are the details that you need to know. Now we move people for the MCQ paper. MCQ paper. That MCQ paper also will start the reverse way. We'll go from people 25. We'll go from 25. We'll come backward. We'll go backward. Reverse is better. Right, reverse way is better. Right, reverse is better. So question number 25. We have 25 minutes, 25 questions, right? 1 minute per question. Which of the following are correct about the seedless vascular plants? Which of the following are correct about the seedless vascular plants? Pterophytes have rhizomes and produce highly dissected fronds. Pterophytes have rhizomes and produce highly dissected fronds. Pterophytes have rhizome and highly dissected fronds. Fronds are leaves, dissected. That is correct, people. We can agree with that. This statement is okay. Lycophytes produce upright stems and ground-hugging stems. Lycophytes produce upright stems and ground-hugging, correct. All seedless vascular plants are homosporous. All seedless vascular plants are homosporous. All seedless vascular plants are homosporous. Seedless vascular plants are homosporous is wrong. We know some are heterosporous like Selaginella. They possess xylem and phloem for transport of water and nutrients. That is correct. And dominant plant is the gametophyte, wrong. So we can take A, B, and D as the answer for that question. A, B, D. Moving to question number 24. Which of the following are true for non-vascular plants? True for non-vascular plants. Haploid gametophyte is the dominant phase of the life cycle. Haploid gametophyte is the dominant phase of the life cycle. Haploid gametophyte is dominant phase of the life cycle. Gametophyte is the dominant phase of the life cycle. Non-vascular plants, correct. Male plants produce flagellated sperms for fertilization, correct. And then all have haploid gametophyte is the dominant phase of the life cycle. Non-vascular plants, haploid gametophyte is the dominant haploid gametophyte is the dominant phase of the life cycle, correct. Male plants produce flagellated sperms for fertilization, correct. All have rhizoids to absorb water. All have rhizoids to absorb water. Which of the following are true for non-vascular plants? All have rhizoids to absorb water. That is sporophyte stage, wrong. Sporophytes have an independent existence, wrong. And then they are found in specially common in moist terrestrial places, correct. So we can take people which of the following are true. A, B, E. A, B, and E. A, B, E. 24th. A, B, A, B, E. Right? Do they ask false or which of the following is true for non-vesicular plants? Right? True for non-vesicular plants. Right? True for non-vesicular plants. Haploid gametophyte is dominant phase. That is correct. And then the male plant produce flagellated sperms for fertilization. That is also correct. Male plants are producing flagellated stages. Correct. All have rhizoid to absorb water. That's they have rhizoids, right, to absorb water. But saying people all is not very clear. Yes, sporophyte and the gametophyte. Sporophyte is not having any rhizoids. If they had like mentioned all gametophytes have rhizoid for absorption of water, we can agree with that. Then sporophytes have independent existence. No, it's depending. They are found especially in common moist terrestrial places. That is correct. So ABE must be the answer. ABE, right? ABE must be the answer. C and D are wrong. Right? Question number 23. Question number 23. Question 23. Which of the following are characters of C4 plants? Following are characters of C4 plants. They concentrate carbon dioxide around Rubisco enzyme. They concentrate carbon dioxide. That is correct. They have Kranz anatomy in their leaves. Correct. Bundle sheath cells enclose vascular bundles. Correct. And then they are more efficient than C3 plants in hot and dry condition. Correct. PB carboxylase is less efficient than Rubisco. Wrong. Then A, B, C, D, A, B, C, and D must be the answer. Moving to question number 22, which of the following statement are correct about Calvin cycle? Correct about Calvin cycle. Take place in stroma of the chloroplast. Calvin cycle, stroma of the chloroplast, correct. And then the first stable product is 3-phosphoglycerate, 3-PGA, correct. The cycle must take place 3 times to produce 1G3P, correct. 3 times, ne? Carboxylation, reduction, and regeneration are the 3 main steps. 3 main steps, correct. And then it requires NADPH but not ATP. Wrong, ne? They're both needed. Inang A, B, C, and D. A, B, C, D must be the answer once again. Question number 21. Which of the following statement are correct regarding photosystems? Photosystem 1 is known as P700 and absorb light at 700 nanometer wavelength, correct. Photosystem 2 contain chlorophyll A molecule called P680, correct. Photosystems chlorophyll contains chlorophyll A molecule. Metana podiya ulla tina pe molecule the 2 molecules dekak tino neda. Photosystem 2 contain chlorophyll A molecule. We'll just leave that. I have an issue with this statement, right? But we'll see. We'll go for the best answer. Photosystem consists of reaction center complex and light harvesting complex. Photosystem consists of reaction center complex and light harvesting complex, correct. And then in PS1, the chlorophyll A molecule is known as P700, correct. Light harvesting complex consists of various photosynthetic enzymes. Light harvesting complex consists of various photosynthetic enzymes. Wrong. Wrong. So what can be the answer? What can be the answer? Which of the following statement correct regarding photosystems? Photosystems. Photosystem 1 is known as P700. as it absorbs P700. Photosystem 1 is known as P700 as it absorbs light at 700 nanometer wavelength. Photosystem 1 is known as P700. Photosystem 1 is known as, sir, in A, chlorophyll molecule is called P700, not the photosystem, right? That's the point, right? Photosystem 1 is known as P700. P700 as it absorbs light at 700. Photosystem 2 contain chlorophyll A molecule called photosystem 2 contain chlorophyll molecule called P680. B is okay, ne? B okay. Photosystem consists of reaction center complex and lighthouse. C is also correct. And PS1, the chlorophyll molecule is known as P700, chlorophyll A molecule. Right? Within a chlorophyll photosystem 1 is called as P700. The pigment molecule is P700. Pigment molecule. So BCD BCD. We'll take the answer as BCD. Photosystem 1 known as P700. It's not very correct. If you read your book, people, it says photosystem 1 pigment is P700. Right? So in PS1, chlorophyll A molecule is known as P700. Two people comment on that. So that is why it's wrong. So we'll move people to question number 20. Which of the following statement is incorrect regarding members of phylum Lycophyta? Members of phylum Lycophyta. Members of phylum Lycophyta, what they ask people, which of the following statement is incorrect? Right? Incorrect. All spike mosses are heterosporous. Spike moss heterosporous, correct. They depend on external water for fertilization, correct. Their gametophytes are photosynthetic, correct. Selaginella and Lycopodium are club mosses, wrong. They are seedless vesicle plants, correct. So we have the answer now. Fourth one is the answer. Moving to question number 19. Correct statement regarding non-vascular plants. There are some sporophytes completely depending on the gametophyte. Correct regarding non-vascular plants. There are some sporophytes completely depending on gametophyte. That is correct. Sporophytes are bisexual. Wrong. Gametophytes, ne? Some club mosses show heterospory. Club moss show heterospory. Wrong. All are consisting of tracheids for water conduction. Wrong. They don't have xylem and phloem. There is only one female gametophyte develops into megasporangium in spike moss. Wrong, neither. Right? So we have the answer. 19th, people, we can go with the first one. 19th, first one. Right, people, we go now with the question number 18. Lamai, question number 18. Question number 18. Pogonatum. A. Pogonatum. B. Neplephis. C. Caecus. D. Selaginella. E. Lycopodium. Of the above given plants, homosporous plants. Pogonatum, homosporous. Neplephis, homosporous. Caecus not. Selaginella not. Lycopodium, homosporous. A, B, E. A, B, E. 5 is the answer. A, B, E, 5. Question number 16. Question number 17. Homosporous plant group. Bryophyta and Pterophyta homosporous. This is correct. Number 1 is the answer. Number 1 is the answer. Question number 16. All the members of the phylum Lycophyta All members of phylum Lycophyta. All members of phylum Lycophyta possess adaptations for terrestrial life. Do not produce heterospores. Possess adaptations for terrestrial life. That is correct. Do not produce heterospores. Wrong. Need no external water for fertilization. Wrong. Produce seeds without fruits. Wrong. Photosynthesize only with sporophyte. Photosynthesize only with the sporophyte. Wrong. So we'll take first one. It's not fully adapted, but some adaptations are there for land life. Non-vascular seeds, non-vascular and seedless plants. Non-vascular and seedless plants. Netam. Non-vascular, seedless, non-vascular and non-vascular plant. Non-vascular plants, you can take Anthocerus. Anthocerus, ne? This is better, ne? Seedless, anyway, seedless, ne? Right? Anthocerus. Next one, question number 14. Which of the following seedless vascular plants evolved closest to the seed plants? Closest to the seed plant. Neprolephis. And then people, question number 13. Which of the following can be found in sporophyte of anthophytes? Sporophytes of anthophytes and sporophytes of bryophytes. Which of the following can be found in sporophytes of anthophyta, flowering plants, ne? And sporophytes of bryophytes. Sporophytes of anthophytes. Shall we take stomata? I think we can take stomata as the answer. Here, stomata, because it's present in the sporophyte of bryophyta. Can be found in all terrestrial plants. Can be found in all terrestrial plants. Reduced gametophyte. Correct. Ovules. Ovules, not in all plants. Ne? Nourishment of embryo, that's correct. Heterospory, wrong. Stomata, wrong. This is also wrong then. Ne? Answer is third, because all terrestrial plants reduce gametophyte, not for Pogonato. Ne? So third one is correct. Third is correct. Question number 11. All seed plants begins. All seed plants do not contain flagella. All seed plants do not contain flagella. All seed plants do not contain flagella. Show heterospory. All seed plants do not contain flagella. Wrong. Ne? Some Cetus has. Show heterospory. Correct. Contain sieve tubes. Wrong. Sieve tubes. Contain sieve tubes. Sieve tubes. Sieve cells. Ne? Sieve tubes are present in netum and anthophyta. Ne? So contain sieve tubes, not correct. Contain ovary, wrong. Deliver male gametes into the ovule by pollen tube, wrong. So we'll take second as the answer. Second as the answer. Question number 10. Select the correct answer based on the following statement A and B. Correct answer based on A and B. A. Gymnosperms are evolutionary closest to pterophytes. Gymnosperms are evolutionary closer to pterophyta rather than lycophyta. That is correct. That statement is correct. Gymnosperms are seed-bearing vascular plants. That is correct. So both A and B are correct, and B does not contribute to A. Closer to gymnosperms are seed-bearing vascular plants. Yeah, we'll take fifth one as the answer. B does not contribute to A neither, because the statement B does not give any contribution to the statement A. Question number 9, Lamai. Which of the following comparison between C3, C4 photosynthetic plants is incorrect? C3, C4 photosynthetic plants incorrect. What do you want to say, people? Incorrect one. C3 carbon dioxide fixing occur once. Correct. Occur twice. Correct. Major carbon dioxide is REBP. C3 acceptase PEP. Correct. Carbon dioxide fixing enzyme is REBP carboxylase. Correct. RBP carboxylase oxygenase. Anyway, carbon dioxide fixing is by PP carboxylase. Photosynthetic yield is high, wrong. Yield is low, wrong. So fourth one is wrong. First product of photosynthesis PGA, oxaloacetate, correct. Fourth one is the answer. Moving to question number 8. Question number 8. Question number 8. Which of the following statement is correct regarding absorption spectrum and the action spectrum? Question number 8. Absorption spectrum and the action spectrum. The effectiveness of photosynthesis is 500 to 600 range of visible light. Wrong. This is green color light. Is not effective. Photosynthetic happen at maximum 600 to 700 wavelength range. Blue light also. Carotenoid pigments absorb. What they ask people? Correct. Photosynthesis happen at maximum at 600 to 700 wavelength range. Carotenoid pigment absorbs only 600. This is wrong. Chlorophyll A highly absorbs 600 to 700 wavelength range of visible light. Chlorophyll A absorbs 600 to 700 wavelength of visible light. Correct. Chlorophyll A absorbs more at 600 to 700 nanometer wavelength range than chlorophyll B absorbs. That one graph, can you check that? People, how that graph changes? Chlorophyll A absorbs more in the blue region, less in the red region. Chlorophyll B less in the blue region and more in the red region. Am I correct? Check that graph, people. Check that graph and tell whether I'm correct or not. Right. So question number 8, we have to select the correct regarding absorption. We'll check this question once again. Right. Correct. Chlorophyll B absorbs more at 600 to 700 range than 400 to 500. Shall we check that? Shall we check it? This graph. How about that? It's the other way around. Blue region, chlorophyll B absorb more. Red region, chlorophyll A absorbs more. So that is wrong statement, right? So we'll take this statement wrong. What happened? We'll take this statement wrong. Chlorophyll A highly absorbs 600 to 700 nanometer wavelength range of visible light. Chlorophyll A highly absorbs 600 to 700. Chlorophyll A highly absorbs 600 to 700. Is it? Compared to this graph, from here, this is chlorophyll B, This is chlorophyll. Take like that. Both chlorophyll A and B absorbs more in the blue region. In the blue region than in the red region. We got to be careful. Chlorophyll A highly absorbs. Wrong. This is also wrong. Carotenoid pigments only absorb 600 to 700. Wrong. Photosynthesis happen maximum at the range of 600, 700, wrong. Effectiveness of low in, this is correct. Ne? So that's the first one. First one is okay. Ne? Effectiveness of photosynthesis is low at 500, 600. That is green region. Ne? Correct. So first one is the answer. First one is the answer. Question number 7. Question number 7. Question number 7. Question 7. Which of the following statement regarding photosynthesis is correct? Rubisco enzyme is efficient only in C3 plants. Rubisco enzyme is efficient only in C3 plants. Rubisco enzyme is efficient only in C3 plants. Wrong, neither. C4, right, people? PEP carboxylase is present in both C3 and C4 plants. PEP carboxylase is present in both C3, C4, wrong. In C4 plants, bundle sheath cells have reduced thylakoid in the chloroplast. Bundle sheath cells have reduced thylakoids in the chloroplast. That is correct. C4 plants, carbon dioxide is fixed in the cytosol of the bundle sheath cell. C4 plants, carbon dioxide is fixed in the cytosol. C4 plants, carbon dioxide is fixed in the cytosol of the bundle sheath mesophyll cells. Wrong. Enzymes are involved in decarboxylation step in Calvin cycle. Enzymes are involving in decarboxylation step in the Calvin cycle. Decarboxylation step. That's wrong. The Calvin cycle is carboxylation, not decarboxylation. Carbon dioxide is added. So third one is the answer. Question number 6. The light-dependent reaction of photosynthesis. Cyclic electron flow occurs in photosystem 2. That is wrong. The cyclic electron flow occurs in photosystem 1. Both linear and cyclic electron flow produce ADP and NADPH. Wrong. Primary electron acceptor of photosystem 1 reduces NADP and yields NADPH. Primary electron acceptor of photosystem 1 reduces NADP and yield NADPH. We'll leave a question for third one. We'll check the other statements. Photosystem 1 gets electrons from splitting of water. Photosystem 1 gets electrons by splitting of water in the linear electron flow. Photosystem 1 photosystem 2 does water splitting. And excited electrons at the primary electron acceptor of photosystem 1 pass through electron transport chain to photosystem 1. Excited electrons from primary electron acceptor of photosystem 1 does not pass to photosystem 2. From photosystem 2, it passes to 1. So people, out of these answers, third one is the best answer. Primary electron acceptor of photosystem 1 reduces NADP. It is not directly the photosystem 1 is involving in NADPH production. There are some more electron transport proteins are engaged. But anyway, this is a process happen, people, together with photosystem 1. So we'll take the best answer as that one. Third one as the best answer. Right, moving people to question number 5. Correct statement regarding C4 pathway. Initial carbon dioxide is fixed by PEP in the bundle sheath cells. Fixed by PEP in bundle sheath cells. That is wrong. PEP in the mesophyll cells. Fixed carbon reaches to the bundle sheath cells as malate, which are more stable state. Fixed carbon dioxide reach to the bundle sheath as malate or aspartate. Correct. Calvin cycle take place in the mesophyll cell of this pathway. Calvin cycle take place in the mesophyll cells of this pathway. Calvin cycle occurs in the bundle sheath cells. Wrong. Regeneration of PEP is a passive process. Regeneration of PEP is a passive process. No, ATP energy needed for reformation of the initial carbon dioxide receptor PEP from pyruvate. And then primary product of this pathway is 4-carbon carbohydrate oxaloacetate. Primary product, the first stable product. Enang answer ka avilla second. Second is the answer. Question number 4, right? Question number 4, which of the event of photosynthesis is least significant to the thylakoid membrane? Which event of photosynthesis is least significant for the thylakoid membrane? Synthesis of ATP, synthesis of NADPH, release of oxygen due to photolysis of water. Which event of photosynthesis is least significant to the thylakoid membrane? Synthesis of ATP happen on the thylakoid membrane, right? Synthesis of NADPH occur on the thylakoid membrane. Release of oxygen due to photolysis of water, PS2. Involvement of electron transport chain. Utilization of NADPH. This happen in the stroma during the Calvin cycle. So we'll take fifth one as the answer there. Fourth answer is 5. Question number 3. Which of the following statement is correct regarding cyclic photophosphorylation? Cyclic photophosphorylation, correct one. ATP can be an end product of B. What is that? Cyclic photophosphorylation. ATP can be an end product. Sorry, this is separating. ATP can be an end product. Cyclic ATP can be end product. Hurry. B. It can occur in bundle sheath cells of C4 plants. It can occur in bundle sheath cells. Bundle sheath cells, wrong. Cyclic, no. Cyclic, possible. Bundle sheath cells, possible. Why? We learn that it happens in PS1 available. PS1 present. So it can occur in bundle sheath cells of C4 plants. Cyclic electron flow, correct. It can be induced by PEP carboxylase in C4 plants. Can be induced by PEP carboxylase in C4 plants. Can be induced by PEP carboxylase in C4 plants. Cyclic, wrong. And D, it can occur in PS2 photosystem. Wrong. It doesn't happen. Photolysis of water can regulate cyclic, wrong. A and B only. Right. Moving to people, last two questions. Which of the following correct regarding photorespiration only occurs in C4 plants? Photorespiration only occurs in C4 plants, wrong. Occurs in C3, but sometimes little can happen in C4 plants. Increase productivity of photosynthesis, wrong. It is a process. Rubisco reacts with oxygen instead of hurry. Third is the answer. It takes place when carbon dioxide concentration is high, wrong. And it is not affected by high temperature and dryness, wrong. Third one is the answer. And the last question in C3 plants, fixation of carbon dioxide involves Rubisco. Rubisco is the answer. Right. So first one is third one is the answer. So thank you very much, people, staying with me tonight, right, to discuss the paper. I hope that you bring this paper on Saturday. I'm going to have a look, right, and then I'll be giving you paper number 7 on Saturday, plus this marking scheme I'll give you on Saturday, right? So thank you, and I wish you all the best, for all people who are tolerating this tough schedule. They make their success, and that's people the truth. That's the truth, right? The practice make perfection, like that. Your hard work, people take you to the success. So trust that, right? Don't keep any doubt on that, and you are going to be winners one day, right? That I guarantee you, right? That you're going to be winners, right? Just by staying and then people working and then practicing, this part is going to take you to the success, right? So congratulations for that. And then I'll be meeting you people on Saturday. I'll be meeting you on Saturday once again, right? On Saturday for our regular class. Then good night. Bye-bye. See you.